So behind my title, Root System Architecture Beyond Geometry, I assign myself another task, which is try to convince or illustrate the benefits or the potential of multiscale modeling in addition or in combination with phenotyping to create useful information. And this was made possible thanks to a very interesting collaboration of Guillaume Lobet, who is a plant modeler, Mathieu Javot, who is a soil physicist and a soil plant modeler, François Chaumont, who is a molecular biologist working on aquaporine expression, and myself, more crop physiology and genetics. So we usually consider root architecture as a question of matching the distribution of root length with the distribution of soil water. And I just would like to make a warning. This is probably much more complicated than that. That usually comes at the last question that people ask, is probably distribution of length, surface, root strength, root exudates, root uptake, root elongation, branching, root hydraulic conductance. All those parameters that are actually distributed at different places around the root system, along the roots, depending on root age, their developmental stage. And we want to have a match between that on one side and the array of parameters that characterize the soil environment. So not only the soil resource, but also soil structures or microbes, the microbiome, and we would like to maintain the match in time and in space. So that's going to keep us for a while, of course. Now, we all know that root systems are difficult to observe, and so the phenotyping is in itself a challenge. And so the root community has developed an array of strategies to try to work with that. And one strategy that has been quite successful is to start from the knowledge we have, our current understanding of how a root system works in its environment. So we start from crop physiology, plant physiology, soil science, to write this down into mathematical equations that allow us to simulate, for example, a root system architecture. Then play with the parameters of those equations to simulate a number of, in this case, root architecture that will allow us then to compute, for example, the amount of overlap between roots depletion zone, which basically is a non-efficient use of carbon. And from this simulation exercise, we isolate potentially useful traits for breeding. The next step will be to validate this, to verify whether there is genetic variation, heritable genetic variation for this trait. Then once we have interest in line, to do an experimental validation that the trait actually is useful. And the loop will start again. So it's a two machine. It's at the same time a trait discovery machine and also a learning process, because the genetics will generate new genetic resources that can be very useful to progress the basic discipline that are behind. And in addition to the new trait, every time there is a trait, it comes with a new phenotyping tool that goes with the trait. Sometimes the phenotyping tool comes before the trait. It's an interesting process. So working that way in terms of water, nitrogen, phosphorus has led to defining, that was progressive over 15 years' time, an ideal root system that would be very efficient to capture nitrogen or to capture water, depending on. And so what I would like to discuss is going back to the loop and trying to go further to this ideotype, which is basically essentially centered around root architecture and geometry. We have an array of technology that have been designed, going from MRI, computed tomography, aeroponics, rhizotrons, and then techniques that are working in the field like chevelomics, and more recently laser ablation tomography that at a very high, a reasonable throughput allows to get the internal structure of the root. And that will become useful later in my talk. So now going back to our current understanding, so I'm the top left of the loop. I'm taking water as an example. The theorem, I like to look at crop models because they sort of summarize our understanding of what works at the field scale. So the potential uptake of water by a crop is usually defined by splitting the soil profile in a number of layers, and in each layers represent the amount of the volume of soil which is basically occupied by roots. And then you take these occupancy factors, you multiply by the amount of water that can be actually extracted by the soil, and you multiply that with the conductance really integrative parameter. So when you look at this equation, you have the feeling that this is a matter of root architecture, and then this would be probably a soil stuff. But basically, this conductance term, which is very integrative, is as much soil as it is plant, actually, because we have the root conductance that occurs. So this dissection is probably fine to work in a crop model. It's probably not the best way to dissect uptake if we want to go to a deep phenotyping. So then I stay still in the top left of my loop and just want to illustrate the limits of the architectural view or the geometrical view. So if we follow the path of water from the bulk soil to the shoot and the evaporation site, we basically see, and this is something that everybody knows or should know, that water is flowing into a bulk soil where the conductance is actually depending on the soil water potential. So, and it's nonlinear relation here. Then it crosses the rhizosphere where the hydraulic properties are likely to be very different. One because water is accelerating when it comes close to the root, and also because we have these root soil contacts, exudates, gaps, air gaps, that makes this compartment very complex. Then it crosses the roots. It becomes a matter of the composite transport model of water flowing through tissues. Then it goes into the xylem, and into the xylem we are going to talk about xylem conductance. But the problem we have there is not the problem, but the heterogeneity comes from cavitation and also the development with the change of diameter of the xylem. And we end up in the shoot, and I'm not going to talk about the shoots. So the plant breeding scope is actually much larger than only the root architecture if we are able to look at those processes. So and now I'm moving to the model, trying to put that into equations. So we use a model, and there are several on the market, they're for free. Model like a robotic that will generate a plant mockup, a virtual plant, so only the geometry. Then we have a model that simulates the water flow in a variably unsaturated soil. And we couple the two by saying that every small position in the soil, water is leaving the soil to enter the root, or leaving the root to enter the soil. That's also possible. And we define then a new model that was introduced by Claude Toussaint, where we have a fully connected soil, plant, 3D plus time model. So what the sort of things that come out of this model, so we start from root architecture. We start also from a map of root water content, and the interesting thing is that these two are things that we can get from phenotyping platforms to some extent. Then there is a direct relationship between the soil water content and soil water potential, from which we can infer the flux of water within the soil. Now, solving the two equations together, we get a very interesting variable, which is xylem water potential, everywhere along the root system, which somehow is the level of water deficit sensed by every root, not water deficit, but water potential sensed by every. So an apex here is feeling much more water depleted than an apex which is located downwards. And then we end up, we also have a magic variable. I call it magic variable because we are probably always wrong when we interpret the maps of soil water content. Is what the soil physicists call a sink strength, which is basically where water has actually left the soil to enter a root. So that's a distribution of root water uptake. And you see that in this situation, root water uptake actually occurs probably, well, it's obviously around roots, but not essentially not specifically the route we would expect to be the most active. So some prediction from, now we are simulating, so we will play with the different parameters. And in this case, we are playing with the ratio of the radial conductance of a root segment to the axial conductance of a root segment. So the axial conductance is basically Xylem, number in Xylem diameter. And we can get those data now through the laser ablation tomography. And the radial conductivity, the radial conductance is a matter of root diameter, but also aquaporine expression, development of apoplastic barriers, and there are more and more molecular markers available to actually monitor this. So what the model says is actually if you decrease the ratio of radial to axial conductivity, or you increase that ratio, you change completely the distribution of water uptake. So if you decrease the radial conductivity, you see that the water uptake as a function of root depth, while soil depth, tends to be constant, while if you increase the ratio, you see that the water uptake occurs mostly on the top profile. Which means that when roots act on or regulate aquaporine expression, and there is already a diurnal cycle for this, it can have this sort of effect to change the position in the soil where water is actually taken up. We probably wouldn't have thought about that. Another consequence, if you look at the Xylem water potential, so that other variable that is generated by the model, you see that in conditions of low radial conductance, you have a Xylem water potential which is kind of stable at every profile. So all the roots are feeling a little bit the same water potential. While if you have a large ratio, then you see that the top roots are experiencing a very low water potential, and while the other ones tend to be unaffected. And this information is interesting because we can relate it, we should relate it with the reaction of the response of elongation related to the soil water potential. And so we know that the roots tend to maintain their growth under conditions of decreasing water potential. So it could be an effect of this, because you see that this situation will tend to reduce root growth in the top surface, in the top profile, and to maintain growth downwards. It could be just a hydraulic process. Now going back to phenotyping, the sort of experimental design that can be used to play with this information is, for example, like transmission imaging, but there are other ways to monitor the soil water content and to overlap this with the root architecture. So this is a map that shows you over 24 hours the change of soil water potential under a maze plan. So from the beginning, the root is occupying the complete root system. So it's not, what you see there is not root growth. It's really water depletion, and it starts from this position and then gradually extend to other parts. So this is what we can get with the model. So obviously, root architecture, we have it. Transpiration, we measure it. The unknown variable here was the Xylem and the axial and the radial conductance. Axial conductance, we estimated based on Xylem diameter. And then the only variable which we were left with was the radial conductance. And so we have been doing what we call inverse modeling, is trying to adjust and find the best value of the radial conductance, which then fits best the simulation results with the observed results. And you can see that it's not that bad in terms of output. Then we can go back to that very interesting variable. Where are those plants taking up water? And from this, you would say, well, they are taking up water just here at the base of the root system. No, from the very beginning, they are taking up water everywhere. But you only see the difference in terms of water content at some places, and this is because the soil is also redistributing the water. And this is something we don't see, because it doesn't involve a change in mass of the complete rhizomes. Okay, now if you want to play with this sort of model, Félicien Meunier has simplified greatly the equations, and Guillaume Lebet has put that into a nice interface. So you can generate all sorts of root system architecture, all sorts of parameters, and see the output in terms of radial flux, xylem water potential. The tool is available online. Just look for Marshall and Lobet, you will find it. And it's an interesting collaboration between Louvain and Yulki. Now, I'm moving to the next step of my loop. We have learned that the radial conductance was probably something to look at. Now, what is behind the radial conductance? So behind the radial conductance, we probably have the root structure in itself. Water has to cross all those tissues. We certainly have the development of apoplastic barriers, where water has to flow from the apoplast to the symplast. And we also have the aquaporin expression. We believe with those three, we should have captured most of, so what is missing is solute transport. Okay, if we want to talk solute, it will not be here. So Valentin Couvreur and Marc Faget have designed a model that has an explicit representation of the root cross-section. So this is a digitalized version of a maze root using a software from Nottingham. And so at every compartment, at every cell position, we have membranes, we have walls, and we have plasmodesmata. And the map of membrane conductance is based on aquaporin expression. The plasmodesmata, we take that from the literature. And well, the cell wall comes from the model. And so we are able to follow the flow density along the walls, across the membranes, and through the plasmodesmata. I can't tell more about this story because it's a very long one, but the sort of thing we can do is to use, that's a simulation step, to play with the parameters. So for example, if we play with the conductivity of the plasmodesmata, we see the effect on the radial conductance. And here we have a situation of development where the endodermis is suberized and we have an exodermis and a Casparian band. That's a fully developed root. Here we have only the endodermal Casparian band, so we are very close to the meristem, elongation zone. And here we have the endodermis suberized with a 3 Cassat cell, which is intermediate. And we see that the contribution of plasmodesmata is essentially important in the more mature part of the root, where the contribution of aquaporins is more important in the young part of the root system. Again, Valentin and Guillaume have been developing a tool online that you can use to play with that, and you can load a number of, we have millets, Arabidopsis, maize, and a number of different cross-sections that can be used for that. And then you play with all the parameters and you see the output. Now, the last step is flowing that into a multiscale, so integrating things at the radial level with the architecture and with the whole plant, not the whole plant, but the whole root compartment functioning to address something that is a nightmare for people doing root phenotyping, which is root plasticity. Because this is a nice root system growing in rhizotron, everything was uniform, but definitely something occurred somewhere, and we had a complete redistribution of that. So trying to understand and to capture this plasticity is something that becomes feasible with the phenotyping platforms. And then how do we interpret that? So that comes from the thesis of Beatrice Moreno and 16 Passot in Montpellier. And they have been studying, if everyone looking at root has seen, this is the case in maize, along a root you have small laterals and long laterals. So because if you take into account that they grow and they are formed in acropetal sequence, the short one is a slow root and the long one is a fast root. There is no way to escape. And so what 16 and Beatrice have been doing, and thanks to them for doing it, is to monitor the growth rate of individual lateral roots for hundreds and thousands of lateral roots. And they come up with this sort of graph, the lateral root elongation rates. Each line is one lateral root. And after some statistical analysis, they propose to split the population in those lateral roots that grow slowly at the beginning and stop growing, those that grow not so slowly and keep growing, and those that tend to accelerate. And there is an interesting correlation between the pattern of the behavior and the diameter, and along with the diameter comes indeed a different root structure. So what we have done is to combine all the different tools that we have, taking the root architecture that is phenotyped in rhizotrons, to digitize that and to look at the root development dynamics so that we can parameterize a root architecture model and simulate the geometry of the system. Then taking transversal section within the roots, 16 and Beatrice have looked at the root anatomy. Then we have been using cell sets from Nottingham to digitalize this and make explicit cross-section hydraulics that yield the KR variable. And finally, using the root anatomy and exciting size, we have estimated axial conductance. And the three information has been input into the larger scale model airstreams. So the lot of things that show up is here you have the water uptake by the fast-growing laterals, the ones that are the most costly for the roots. And you see that if you increase, there is a benefit going to 15%, but after 15%, there is no real reason to invest more in those fast-growing laterals. And actually, the observed range mean in a panel of lines that have been phenotyped by 16 in Nottingham, you see that you are in something that fits quite closely with the prediction of the model, which is always interesting. And then the second is we are trying with people in Montpellier to make the link of that root compartment with the shoot behavior. And so an important variable is the water potential and the color, because this is what the shoot part is actually sensing and to what it should react in terms of stomatal conductance. And what comes up here, so the reference, which is the observed scenario or in terms of proportion of actual of fast growing, is you see over time variation. So that's night, that's day, that's night, transpiring, decrease of water potential, increase. Okay, that's a purple line. If you try to make more fast-growing laterals, you are going to the green, which is essentially the same. If you try to decrease the proportion of lateral of fast-growing, you are going to the blue. And the most interesting one is the mean. If you try to say that all laterals are growing at the same rate, which is average rate, then you get the worst situation. So actually the plasticity seems to be, or not the plasticity, the instability seems to have an added value in terms of efficiency of water uptake on the long term, because this is several days experiment. So to conclude, so obviously you have understood that our idea is that multiscale modeling can be a game changer in root phenotyping. So one message is that root geometry alone captures a rather limited part of the soil root hydraulics. One can identify critical variables at different scales. So the loop that has been running for a while can still run for a few circles before we are done. Combining soil hydraulics, root hydraulic architecture, and root anatomical architecture yield meaningful properties and behaviors. For a number of those model, analytical solutions are available, and then would make this compatible with crop models. So it might be possible to integrate those sort of models into crop models for large-scale simulation. And this is a learning process, of course. One of the problems is that many of the variables at the micro scale are just not observable. And so it's always a question when you try to come up with those studies, there will be someone telling you, okay, but you have not validated. How do you want me to validate the flux of water within the cell wall and the third cell layers in the roots? That's not possible. So if we want, but we want to progress, and we will progress in more. And finally, I think, but that's for the next generation of researchers, we need to extend this to the rhizosphere and to more complicated things. We cannot escape that. And maybe just a slide. So through EPPN 2020, you can access 14 times, not 40 times for one group, the aeroponics phenotyping facility in Louvain. I tend to consider aeroponics as a way to observe roots in non-constrained environments. For there to say that it would be a good reference, there is still a gap, but we use an array of techniques to monitor root growth with a time resolution of 2 hours. So we have 1,000 plants, and every plant is imaged every 2 hours. So that generates a lot of images. And so the link is easy. That's our university site /aeroponics. And you are welcome to discuss. There is a poster for it. Thank you very much.